Thank you, Tyler. Never had an introduction like that before. You know, I got in last night and I was at the hotel and I was working on my slides, but I had my iPad next to me and was kind of reading stories about Steve Jobs, you know, and it's just like so profoundly sad, you know, that he's at the pinnacle of his career. You know, he's created all these products that influence our lives every day, and you know, and he passes early at a fairly young age. And so, as Tyler said, you know, earlier in my career, I wanted to switch. You know, I just felt like the area I was in wasn't having much of an impact, and so I made this fairly big jump to get into life science research. And you know, you hope you can make a difference. And so today I wanted to talk to you about some of the recent things we've been doing at Caliper, and in particular we've been working on doing multimodality imaging, integrating optical and CT imaging. And probably, as most of you are well aware, optical imaging really has become quite powerful recently, and in our area of interest in particular is molecular imaging, and so there's all kinds of different forms of this. We have things, for example, like you can take cells and animals and you can engineer them to express the luciferase gene, and this, when combined with ATP and oxygen, emits light, so you can use that to track the growth of tumor cells or stem cells or bacteria. We can do the same thing with fluorescent proteins. We can label antibodies with fluorescent dyes and use those to target different things. And then there's a whole class of nanoparticles, quantum dots. So this overall area of molecular optical imaging has really taken off over the past few years. And you know, some of the strengths of optical imaging, one is just huge dynamic range. On the imaging systems we develop, we have a dynamic range of about 10 to the 11th. I don't think there's any other modality that has that kind of range. Excellent contrast, at least in the case of these genetic reporters. Quantitative in the sense that signals proportional to the number of cells or number of molecules. Genetic reporter, and probably more relevant to you all, clinically relevant, at least in the area potentially of dyes and nanoparticles. This just a sequence of photos or images here just shows a tumor model, a subcutaneous tumor model, where we can follow, you know, this tumor signal from on the order of one cell all the way up, you know, through 42 days. And over here we've quantified the signal. You can see, you know, here's 10 to the 4th, 10 to the 10th. It just shows sort of the amazing dynamic range you get with optical imaging versus calipers, for example, where you can't you know, measure the size of the tumor for the first couple of weeks, and then it jumps up at the very end here. So that you know those are some of the strengths of optical imaging, but the challenges, as you all well know, are that photons are absorbed and scattered in tissue. So we have a source somewhere in tissue, and we're really imaging the light coming out on the surface. And the photons that you detect on the surface depend on the source depth. And there are tricky things that go on here, like photons generally follow the shortest path to the surface. That's where the signal intensity is largest. And so if you look at this is a phantom here, but we've done an X-ray image of this phantom with a rod, and the light source is at the end of this rod. And then we do an optical image, and you can see the light is off to the side of the rod, and that's because here's a cross-section right through here. And the light is taking this path to the surface, and the X-rays are taking this path. And so light propagates in ways that aren't, at first, intuitively obvious. So this makes quantification challenging, it makes resolution challenging, and there's no anatomical information in this optical image. And so this is well known, and we've been wanting for years to start combining optical with anatomical imaging modalities. And so we've been working recently with CT in particular, And this image over here on the right shows a CT image with contrast agent. You pick up kidneys and so on, co-registered with a, oh, very nice. I need that for these CT images. Co-registered with a 3D reconstruction that we'll talk about of a bioluminescently labeled prostate tumor. And so now, you know, you have localization, you have quantification, and you have anatomical context. So we're going to talk today a little bit about how we've been doing this sort of imaging and a little bit about how we do the tomography. We have a couple terms we use. One we call DLIGHT, which is diffuse luminescent imaging tomography, really applied to bioluminescence, and flight fluorescence imaging tomography. And they use, you know, they're both based on diffuse tomography, but they use very different methods for getting the reconstruction. And why CT imaging versus, say, you know, other methods? Well, you know, first of all, it's a relatively simple and low-cost way to get anatomical information compared with, say, MRI, which would also be nice. And one of our interests is we really want a good measure of the surface topography of the animal, which is important for our optical reconstructions. You get the bone, obviously. You can get soft tissue, and especially if you use contrast agent. And one of the key points is that some of the new CT designs, new detectors have, you know, enable low dose during the imaging, which for us is critical because that's the whole point of optical imaging is you can do longitudinal studies. So if you want to combine it with CT, you want it to be low dose. You can also do the CT imaging in a longitudinal manner. And down here just are miscellaneous images from CT systems just showing the kinds of the kind of work that can be done. So we've been into this area for a few years now, and this slide just highlights some of the instruments that we're combining, where we're combining X-ray and optical. This Lumina XR is a high sensitivity 2D optical imaging system for bioluminescence and fluorescence, and it has a 2D X-ray imaging capability. And then about a year ago, a little more than a year ago, we introduced a CT system called Quantum FX. That's this on the right. And we have our IVA spectrum system that does the optical tomography. And so we've been combining images from these two systems. And this is a low-dose CT system, and we've developed mouse shuttles and software and so on to enable the co-registration of the two systems. And then what I want to talk about today mostly is this new system we have, IVA Spectrum CT, where we've integrated optical and CT imaging into the same box. Basically have the same performance as the Spectrum over here, and it also provides this fast low-dose CT that is of interest to us. I have just a couple slides on the quantum to kind of set the stage for Spectrum CT. So one of the key features of this system is that it was very fast. It has a continuously rotating gantry, so we can actually do animal scans in as short as 17 seconds up to 2 minutes with a little bit higher contrast. So again, low dose and on the order of 10 milligray per 17-second scan. And one of the key other features is it has 2 resolutions, so we can do whole animal imaging, and then there's a way you basically slide the detector and source closer to the animal to magnify the image, and you can get high resolution images. And then, as I mentioned, we've developed some of these unique co-registration tools. And this just shows this mouse imaging shuttle that we have, so we can put a mouse in here, we can place it in this holder into the spectrum, and we take it out and we drop it in another holder that has a fiducial pattern, and then that goes into the quantum, and we have software that recognizes that fiducial pattern and co-registers to optical. And you can get some nice results with this method, but you know, it remains somewhat tedious to do this. Maybe these systems aren't in the same lab. You know, it's not ideal. But these are the kinds of images we've gotten from this this combination. This is an MB231 luciferase labeled breast cancer cell line with an intracardiac injection, and then we get metastatic lesions forming through the animal. We did a delight reconstruction of these lesions and overlaid them on the skeleton from CT. And now we can say, take these regions of interest, we can quantify those as a function of time, and so now you have this anatomical context, and you have the correction for tissue absorption because we're doing the delight reconstruction. And then you can take it a step further, and you can zoom in, say, to get a high resolution of the bone where you have this metastatic lesion. And now you can see the bone damage that's occurring, and you can quantify, say, bone volume and compare it with bioluminescent signal. So these are the nice sorts of things you can do when you have CT and optical. So now I want to move to the Spectrum CT system, and our goal here was to try to introduce CT without compromising the optical capability that we have, and I think we've accomplished that here. And part of the way we've done this is we've adopted a new CT imaging concept, at least maybe new in the animal imaging world, which is a turntable configuration. So the animal sits on a turntable, and there's an X-ray source, which is located, this is the back of the IVUS system, there's an X-ray source located at the back, and we project a cone beam across the animal onto a CMOS flat panel detector, which is located on the front of the instrument, and then we rotate the animal on this turntable, and you can then do a CT reconstruction from that data. Now you'll notice, first of all, it's a very compact, low-cost method to introduce or to implement CT. There's no big gantry involved. The other thing is it keeps this vertical line of sight clean for doing all the optical imaging we want to do. We have transillumination hardware underneath the animal for doing fluorescence transillumination, and the detection camera equipment is up top. So we've kept that line of sight clear with this CT configuration. So our scan times are on the order of 7 to 75 seconds. Dose, again, as low as 10 milligray. A therapeutic dose is on the order of 1 gray, so we're maybe 100 times below the therapeutic dose. Field of view shown here, and voxel sizes down to 40 microns. This is just a little movie we made showing the concept. So the stage drops down, the X-ray beam comes across, and then we spin the turntable. We can put 2 mice on the turntable as well as one, so we get a little bit higher throughput. And then this next image shows the stage then moves up in the box, and we can illuminate for epi-illumination or do bioluminescent imaging, and produce these sorts of images. This just shows a little more detail of all the optical components we have up at the top here. This is a very large CCD sensor. It's 1 inch square, and so we have very, very large lenses and filters and so on to support that. So the CT system has these different operating modes. We can do what we call a coarse scan. If we just want the mouse surface, this is a very useful scan. It goes fast. We can actually do this coarse scan down to 3.5 seconds. Reconstruction time very fast, but the resolution is sort of marginal. But we have these improved resolution modes all the way down to a higher resolution state here. And I'll point out that these CT reconstructions are done using parallel processing on a high-performance GPU card, and that enables these very rapid reconstructions. And this slide just shows the images for these 4 different operating modes. Coarse scan, you know, a little hard to see, but like the ribs don't quite come out here, but it's nice, you know, bone, nice surface rendering, standard, medium, and fine. So as I mentioned, even though we've added CT, we've tried to keep all these optical capabilities that we had in our original spectrum instrument. We can do in vitro imaging. We have a high resolution mode for looking at insects like this, bioluminescent fluorescent, infrared fluorescence, short wavelength like GFP fluorescence, very high sensitivity. Here's a single cell imaged, subcutaneous. We can do single mouse imaging. We can do 5 mouse imaging for high throughput, and of course the tomographic co-registration. And this slide just shows some of the optical features. One of the key things is we have 18 emission filters and 10 excitation filters. So that was our goal here, is to really completely cover the visible to near infrared part of the spectrum for doing all types of molecular probe imaging. Very high transmission in the filters. And one of the other key aspects of this optical system is our ability to switch from reflection fluorescence imaging mode. So here we have a lamp going through a excitation filter. We illuminate the animal from above and then collect lights. We call that epi or reflection mode. But then we have an optical switch here, and we can switch to illuminate from below. And here we can do an XY scan of a point source below the animal, and we use that, 1, to improve signal to background for fluorescence imaging, and 2, to do the fluorescent tomography, which we'll talk about in a minute. So one of the key things that a lot of people struggle with optical imaging is, what does it mean to be quantitative? And so this slide tries to address some of these issues. We detect light or photons on a CCD camera, and as you know, CCD camera has output in counts, like 65,000 counts for a 16-bit camera. But the amount of counts you get obviously are affected by everything that's in front of the camera, the f-stop, the lens, the animal position, a number of things. And so the first goal is to calibrate or connect counts on a camera to a physical unit like surface radiance. How many photons per second per square centimeter per steradian are emitted from the surface? So that's sort of a first level of calibration. And this value is unaffected by camera settings. They're calibrated out in a sense. But this isn't absolute calibration either, because this signal is affected by how deep the source is, its location. And so to get from surface radiance to, say, your optical source of interest, you need to do some kind of 3D diffuse tomography reconstruction or a calibration to take that tissue absorption into account. And then you get your actual photon source, how many photons per second, say, are emitted from this point, which can be related to the number of cells that are labeled or the picomoles of dye molecules. And that is the quantitative thing that we're interested in the end. So on the tomography part, we often get asked, what kind of resolution can you possibly achieve? And we've done some simulations to try to bound the problem, like what is the best you could do. Here we have a trapezoid shape, and this again is a simulated shape with simulated tissue properties, and we've modeled 2 sources here, and we've changed the spacing and depth of these sources, and then did reconstructions introducing photon statistical noise and that sort of thing. So this represents maybe a best case of the kind of resolution you can expect. And so here's a reconstruction where the 2 sources are resolved. Here's a case where they were separated by 0.5 millimeter and they're unresolved. So we look at what point are these resolved, and we're calling that the resolution over here on the left, so that those are the blue diamonds. But then we can also quantify the absolute intensity of the source from this reconstruction. That's the red points here. So resolution maybe is a millimeter when you're just a few millimeters deep, and degrades to a few millimeters when you're, say, a centimeter deep, which would be like the center of a small animal. So not bad, but not great. But the quantification actually looks pretty good. We're accurate to just a few percent here. And as we go deeper, maybe 5 to 10% accuracy on the quantification. So that's the really key point of why we're interested in doing these reconstructions. So I'm going to talk a little bit about how we do the bioluminescent and fluorescent reconstructions, and they rely on different techniques. The bioluminescent in particular relies heavily on differential absorption at different wavelengths. So as you all well know, you have high absorption hemoglobin and other tissues in the short wavelength range up to about 600 nanometers, and low absorption above that. The firefly luciferase spectrum is shown in orange here, and it nicely spans high to low absorption. So we can actually do imaging at different wavelengths and sample, if you will, light that's coming out with different absorption properties, and that provides a very strong constraint on a tomographic reconstruction. So these are the two different methods for bioluminescence and fluorescence. In the case of bioluminescence, as I mentioned, we'll image multiple wavelengths. So here I've just shown two, and the shorter wavelength will have a narrower peak on the surface and a lower amplitude compared to a longer wavelength. And so we can do multiple of these. And then there's a simple model. We have a source multiplied by a Green's function matrix, which is basically the diffusion model from this point to the surface, and that will give us the photon density on the surface. So we need to solve that equation for the source. With fluorescence, we're going to move the transillumination point on the bottom of the animal here, and we're going to create different path lengths of illumination from this source to this source. And so when we have a short path, we get a big signal. When we have a longer path, we get a smaller signal. And again, we can do many, many transillumination points to gather a big data set. And in here we're solving essentially the same equation, only now there's a coupled matrix here. We have an excitation Green's function and an emission Green's function, but the basic concept is the same. One other thing I wanted to point out is one of the things we've worked very hard on is trying to figure out how to do tomography fast, meaning in a few seconds. And we have a couple techniques for doing this, and one of them is we use what I call a planar boundary approximation, meaning that we treat the full shape of the animal, but on each surface element we approximate that as a local planar boundary. This allows us to calculate Green's functions analytically, and we can compute them in advance if we need to, store them in a table, and so we get very fast computation of the Green's functions versus doing a, say, more accurate thing, which would be like finite element modeling or Monte Carlo modeling, which would be very tedious. And we found that for relatively smooth surfaces, doesn't have to be cylindrical, but relatively smooth surfaces, that this approximation is very accurate relative to other things that you don't know, like the exact tissue properties and heterogeneous tissues and so on. So here's sort of diagrams that show how the reconstruction proceeds. In this case, we have a rod embedded in a phantom. And here I'm showing a luminescent rod. Here I'm showing 4 images at 4 different wavelengths from 560 to 620 nanometers. You can see how the signal increases with wavelength. Then we determine the animal surface from the CT image. We map this light onto this 3-dimensional surface. We call it photon density once we map it onto the surface. And then we solve that photon diffusion equation. Right now we use a non-negative least squares algorithm with regularization. And one of the other key speed things is that we do this adaptive mesh where we start with relatively coarse voxels, and this allows us to localize the region of interest, so we can say throw out voxels up here. Then we can refine the voxel size down until we converge on the final solution. And so reconstruction like this generally takes on the order of 5 seconds, up to maybe a minute for more complex shapes. One of the other, challenging considerations is how do you make sure, especially in our spectrum CT configuration, how do you make sure that the X-ray images are co-registered with the optical, because they're really two orthogonal systems. And so we have to do a number of things. On the optical side, we do this intensity calibration, but we also have to do distortion correction. All lenses distort the image somewhat, and the CT images don't have the distortion, so we have to do a distortion correction. On the CT side, we do a reconstruction of the surface, but then we have to apply a transformation calibration, if you will, that maps that surface to the optical image. And this shows an example of overlaying a photograph underneath, and we've colored the surface here in purple and overlaid it. And so we do things like this to check that we have perfect co-registration. And so then once that's done, we can then take our CT image and we can map the optical signal on there with confidence that they're co-registered. So I'd like to walk through now just some validation studies and real animal imaging examples that we've done in this new system, starting with our favorite phantom. This is where we always begin because we know the optical properties and we know where the source is located and we know its intensity, so it's a perfect validation source. In this case, we have 2 fiber optics that go into this phantom, and we can run them both or turn them on one at a time. We also have fluorescent rods that we can insert into this phantom for fluorescent validation. And this is a great example of the kind of mysteries that you see in optical imaging when you look at sources at different depths. So here is the phantom I just mentioned where the source is somewhat shallow. That's source A, and we do the optical image. Here's source B, which is deeper in the mouse, and this is the image that you get. And it looks like you have 2 sources here, but you don't really. You have one right in the middle, but the light's kind of coming out the sides because that's the short path. If you were to quantify these 2, they're much different numbers of photons per second on the surface, but the source underneath is the same amplitude. So this is the problem really that we're trying to solve. And so here's a co-registered reconstruction of that source A. This is the CT image. These are CT cross-sections. And you can actually see the fiber optic in the CT image, and the tip, the fiber, is right here. And I've overlaid with pseudo-color the optical reconstruction of our source. So this is a point source. Our optical reconstruction is probably on the order of 2 millimeters in diameter here, so that's about the kind of resolution we can get. And so in this case, we had a depth error of 2% and a source intensity error of 13%. So we consider that good, very successful. And here's the source B, which is much deeper. Remember, we're imaging from the top, so the source now is near the bottom of the animal, but we still get all the information we need to reconstruct its location. The errors are slightly larger for this deep of a source, but still very acceptable. We have the same kind of validation for flight, and this just shows a typical data set. The little plus symbols here show the illumination point, and the source is about located here. So you can see when you're illuminating far away from the source, the signal's weak but still present. As you illuminate closer to the source, then it gets very bright and very broad here also. And this is a flight reconstruction of that source. And if you can see, there's in the X-ray image, this is the rod, and there's a hole in the rod right there, and that's where the fluorescent dye is located.
The reconstruction is off just a little bit. It's like a millimeter or so off from its actual location, but you know, fairly close. So moving now to some implants in real animals. This is a nice example because, again, it shows kind of the resolution versus quantification issues. This actually is a luminescent ring that we constructed and implanted at 2 different depths in the animal. And the ring, you know, looks a little bit like a lifesaver. It's a little smaller than that, but it's a kind of a ring shape with a hollow center. When we implant this shallow, we can reconstruct. This is kind of a blow up of the reconstruction. We can see the ring shape in our reconstruction. It's not perfect, but you see the hollow nature of it. As we move this deeper in the animal, so now it's kind of right in the middle, we lose the ability to get the ring shape, but we've actually reconstructed the location and the intensity of the source correctly. This table down here shows the depth, blue being the measured depth. The other bars here show the reconstructed depth, so pretty close, within a millimeter. And most importantly, the source intensity or flux quantification was right on in both of these cases. So that is a nice result. This is a fluorescent bead implanted in an animal with very low amount of fluorophore. This is a dye that emits at 750 nanometers. We have 0.01 picomole in this little capsule, and it's implanted. And you can see our signal here is very, very weak, but we were able to do the reconstruction. There's a small dot here. I don't know if you can see it, but that's our reconstructed point. And again, you can see the capsule or located here in the CT image. So good co-registration there. The other thing we've been working with a little bit is just the benefits of imaging the animal unconstrained versus constrained. This is unconstrained. You can see the cross-section of the animal right through this region, kind of the lung area. Very steep, right? It's very pointed, very, very tall. But mice are squishy, and you can compress them. We have our mouse imaging shuttle has the ability to compress somewhat. Main advantage of doing this is you make the tissue thinner, and because of that you get more signal out. You can see the brightness of the signal here compared with this case. This is the same animal in both cases. And the position of the bead has changed, right, because of the compression. The signal on the surface changed, but the reconstruction found the source and got the source intensity correct in this case. We got 0.008 picomoles versus the 0.01 that was injected, so within 20% or so. So now a few real biological models. In this case, we have this MB231 Luke breast cancer cell line, luciferase labeled intracardiac injection. I mentioned this earlier. 1 million cells, and we can track mets forming. And so here, this is the early stage of this, the progression of the disease in this animal. So we have sort of the first, maybe second met forming here. This is fairly shallow, but we did the reconstruction. And again, it sort of nicely overlays with this region of the leg here. And in the spectrum CT, we also have the ability to zoom in and get higher resolution image of the bone. Can't get quite as high as on our quantum system, but still pretty good. Enough to see the effect or damage of the lesion on the bone here. This is another live animal example where we took an HD 460 Luke lung cancer cell line and injected into the lungs. And here we had 1 million cells suspended in Matrigel and injected into the lungs and did a reconstruction. Again, it's a nice sort of validation case because you can see the lung easily in the CT image, and you can see the luminescent luminescent signal in the lung as expected, kind of spread out, but kind of a nice example. Then this is just a fun movie showing two of these animals with the lung injections imaged side by side, so we can actually, as a higher throughput mode, image two animals at once and do the reconstructions and CT on both of them.
Brad, is there any soft tissue contrast at all from the CT? So do you get some spatial information out, and do you use that as a prior in your reconstructions?
So first of all, this next slide is a good example. Maybe these are fat, obese mice, and I don't know if this comes through very well from where you're sitting, but here you can see the cross sections of these animals. You can pick up the bone, obviously, but if you can see carefully, you can distinguish between fat and other tissue types. So, and there's no contrast agent here. So the answer is yes, you can get some level of soft tissue contrast. With contrast agent, you can get pretty amazing things. And one of the key points I didn't mention earlier, but with the fast imaging, it opens up the possibility of using like human contrast agents that have very rapid clearance in mice. So with the 5, 10 second, 20 second scans, we can use these human contrast agents, make that a little more routine. And then the answer to your last question is we have not used this yet as a prior piece of information in the reconstruction, but that's an obvious next step to do.
But so all the reconstructions that you're showing are really without any spatial constraints, or are you using some assumptions about?
No, they're the only spatial. So the model is, the detailed model of the surface, so complete surface model of the animal, homogeneous tissue inside. So we've done a lot of tissue property measurements on mice. All the different tissue types. This was a big part of Olivier's work when he was with us. And we've kind of found certain tissue types that work well for most locations in the animal. The lighter tissues like muscle, skin tend to have similar properties, and we generally use those in our reconstructions. But it would be better to use a full heterogeneous model. But all these things that we just looked at are with that heterogeneous, with that homogeneous model. One of the things that helps us a little bit is when you look at the path the light has to go through, it generally doesn't go through the entire animal. So you're not like starting at the bottom and going through lungs and kidney and before you get to the top. Generally, you're going through a shallower amount of tissue that is somewhat more homogeneous. Just a couple other final points is if you pick this up or appreciate this, all these reconstructions that were shown were done from single view imaging. We found that we get, with bioluminescence, we're really using multiple wavelengths to get the depth information. So as long as we have good coverage of the top half of the animal, we get quite nice results. With fluorescence, we're in a sense scanning the top and the bottom because we image the top and we scan the source on the bottom. So that gives you very strong constraints on the reconstruction. So you don't have to have multiple views of the animal, but nonetheless, sometimes it's helpful to have additional views, and I'll show some examples where it might be helpful. But we have now in Spectrum CT, we have this optional side viewing mirror assembly, so we can actually get the two side views and the top view in one snapshot. And the X-ray, by the way, goes right through the mirror, and so you can still do the CT image of the mouse in this configuration. And so you can see it gives you some additional information, especially the lower part of the side of the animal here. But here is that mouse phantom with both sources lit up, and we did a reconstruction with just top view, reconstructed to 2 points, and then using the top and side views. And similar results here. If you look at the quantification, when we added the side viewing mirror, we reduced the error, maybe say a factor of 2. So here we had like 16% error, and we reduced it to 3 and 9% error. So, a modest improvement in the reconstruction here. But here's a case that's more complex. We had like a barbell shape that we constructed and implanted in the side of the animal. So there's a source up here and a source down here. And when you just do the top view, the top source in a sense blinds you from seeing the bottom one. It's more a dynamic range issue, that this one is so bright that you can't actually see this one. But from the side view, you can kind of see here, you can see both sources. So this is the top view, and here's the side view. And so in this case, when we did the top view reconstruction, we just reconstructed the top source, and when we did the side view, we got them both. So a little bit of an artificial example, but it shows that in some situations it can be quite helpful to have that additional view. Final thing I wanted to mention, this is a little outside of the CT imaging, but we've been doing kind of a fun thing recently you may have heard about, which is Cherenkov imaging. And the idea here, there's been a lot of talks at recent conferences on this. The idea here is that when you take radio-labeled probes, PET spec probes in tissue, and you get this electron or positron emission in tissue for a short period of time, those particles exceed the velocity of light in tissue, and you get Cherenkov radiation optical emission. And that optical emission can be detected with sensitive imaging systems like ours. So we've been doing quite a bit of this Cherenkov imaging recently. And all of the things that I showed you, such as the tomography and the overlay with CT, that can all be done for Cherenkov imaging as well. One of the things, we've done some collaborative work with Simon Cherry's group at UC Davis, and one of the things we did was sort of a kinetic distribution study. So we injected PET probes, and we did a kinetic series of images, and we did this thing called DICE, which is basically like spectral unmixing on the kinetic curves here. And so we can separate these curves into 3 different regions. These are basically the distribution kinetics for these different regions in the mouse. The green is the kidney area, the blue is brain, and the tumor and bladder regions here in red. So that's kind of a nice new capability that we have that's really just come out in the past year or so. So I'd like to wrap up there and just say that CT imaging is a valuable tool for improving the quality of diffuse optical tomography reconstructions. It provides us accurate surface rendering, gives us anatomical detail for validation and context, and it provides, in some cases, complementary information. It shows bone damage, for example, that you can't see with optical. It allows us to validate better our delight flight models, and shows good quantitative agreement with the CT location. And key point is this type of multimodality imaging can be used for tracking cancer and other diseases in longitudinal animal models. And Bruce, to your point, we think the next step, in the evolution here is now to improve further optical reconstruction by incorporating measurements and prior information, say from the CT, on getting better heterogeneous tissue maps. So I'll stop there and just want to thank the group up at Caliper that contributes a lot to the hardware, software algorithms that we use in this product. Thanks.